prokaryotic cell structures internal to the bacterial cell wall. So we've talked about, we're sort of taking a mental trip from the outside of a bacteria to the inside. We've just burst through the cell wall. Now we're going to get to that cell membrane and then the delicious goo within. Do you guys eat the delicious goo inside bacteria? If you eat yogurt, don't you think you're eating the delicious goo inside bacteria? Delicious yogurt. All right. Are yogurt bacteria gram-positive or gram-negative? Nobody knows, right? How the heck would you know this? I've got some yogurt. We're going to do a gram stain today. Do you guys think you can figure it out? If somebody feels like doing a gram stain of yogurt, I'll make sure to not eat all my yogurt. Okay? All right. Anyway, so prokaryotic cell structures internal to the bacterial cell wall. Plasma membrane, inclusions, nucleoid, ribosomes, endospores. I would put a little asterisk about inclusions. I'm going to talk about them, but I don't think there's going to be a lot of testing about those, okay? So lower your blood pressure when I talk about inclusions. Keep your blood pressure nice and high when I talk about all these others, okay? All right. So plasma membrane. Plasma membrane is the separation of the inside of a cell from the outside of a cell. This is for any cell, or at least any cell that we're going to talk about in any detail. That includes bacterial cells and eukaryotic cells. We both have what's called a phospholipid bilayer with membrane proteins in it. Yeah? Can you guys see the phospholipids here? Yeah, hopefully you guys, if you see a little critter with a head and two little tails, that's a phospholipid. And you can see them all sort of standing in a row here, yeah? And what do we know about the heads? Polar. And somebody said something that begins with hydro. Hydrophilic, yeah? And then the tails. You guys like that sound? Please stop. Okay, I'll stop. Hydrophobic, yeah? So, are we surprised? So, should we do, let me just take about a couple minutes for a brief thought experiment, okay? Do you guys know what a thought experiment is? There's an experiment where there's thinking. Exactly. Nicely done. How did he do that? All right. So, incredible. So, consider a beaker. And here is some water in the beaker, yeah? And then I take I have a pair of molecular tweezers, and I throw in a phospholipid and drop it on the surface of the water, yeah? It's a lipid, so we expect it to float, yeah? How does it float? Does it float like A, B, or C? A. What do you think? It stands on its head. Isn't that weird? Dude, you're standing on your head. What makes it stand on its head? The laws of physics, yeah? The polar interactions, the head likes the water, the tails don't like the water. So it sticks its head into the water, and then the tails stick out, and that's because of molecular interactions of attraction and repulsion that polar things have for water and nonpolar things have against water, yeah? And so if I continued to pour a bunch more phospholipids into my beaker such that I covered the entire surface with a single layer of phospholipids, we can imagine what that single layer would look like. They would all stand on their heads, yeah? Every single one of them. And if I poured more in, they would gradually fill up until they were all standing on their heads together, weren't they? Does that make sense? guys with me here? Right? So now we can imagine a perfect single layer of ridiculous, right? This is like a big cabaret line. Da, 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 da. They're all sort of dancing on their heads. You ever think about these things? Okay. Right? One singular sensation. Do, 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 do. Anyway, sorry about that. I'm so glad that's really putting the hurt on somebody there. Thank you. All right, so now let's take one more phospholipid and drop them on the top of this layer. What's going to happen? Again, here's A, here's B, here's C. How is he going to land? B. And why? Well, because his tail's like the tails of this guy, yeah? You guys with me here? Right? And so what makes him stand straight up? Why didn't he tip over like this guy? No, what makes him stand up is, again, the laws of physics, the laws of attractiveness for hydrophobic things to hydrophobic things and repulsiveness of hydrophilic things to hydrophobic things, yeah? Guys with me? So if we put another layer on there, what would we have? We would have a phospholipid bilayer. Would we have a plasma membrane? Not quite. We don't have any of these proteins in there, yeah? But we would at least have a phospholipid bilayer. And we can conclude then a large part of what holds the phospholipid bilayer together, yeah? What does hold it together? The laws of physics. Hydrophobic and hydrophilic interactions are what just sort of keep it together, yeah? There's no covalent bonds holding these phospholipids together. Okay. All right. We also see in here some other goodies, cholesterol and polysaccharides, lipopolysaccharides, and glycoproteins, yeah? These are the little hexagon things. These are carbohydrates. These things here, these are lipids, and then the blue things are proteins, yeah? All right, so this is sort of a theorized plasma membrane. It is a fluid mosaic. You guys understand what that means? Again, there's no covalent bonds holding these phospholipids together. They can wiggle around side to side within each other, but they're not bound together. Okay. It is a phospholipid bilayer with proteins in it. Peripheral proteins that sort of attach to the outside or the inside. Integral proteins that go across the membrane. So, how many proteins are made out of what? Let's consider one more part about the phospholipid bilayer, which is these proteins, right? What do we know proteins are made out of? They're made out of amino acids. How many kinds of amino acids are there in proteins for all life? 20. Tell me about those 20 amino acids. Are they all the same? How can they be different? Tell me a relevant way they can be different. They're different in their R groups. That's the little side chain for every one. They're all the same across their backbone, pretty much, yeah? And then they're all different in their R group. So, talk about the R groups. How can they be different from each other? Are they hydrophilic or hydrophobic? Trick question. Some of them are hydrophilic and others hydrophobic, right? They've got both bases covered. Some amino acids have hydrophilic side groups and some have hydrophobic side groups, yeah? All right, well, let me ask you this. What about here? We've got this protein going across the membrane, yeah? what kinds of amino acids are sitting right here in the middle of the membrane? Hydrophilic or hydrophobic ones? Hydrophobic ones. What about at the ends of the protein? What kind of, if you were making your protein, right, what kinds of amino acids would you make here? Hydrophilic ones, yeah? Does that make sense? What holds this protein in the membrane? The same chemical forces, yeah? You guys with me here? Okay. I'm so glad we had this discussion. All right. You know what these proteins aren't sitting here, and these phospholipids aren't sitting here by magic, yeah? As long as you accept hydrophilic and hydrophobic interactions, it makes sense, yeah? All right. So, yeah, so more stuff. Proteins move, and what do these proteins do? This protein says pore, yeah? What do you think this pore does? Lets stuff in and out, yeah? So really, the sophisticated work of the plasma membrane is done by the proteins within it, yeah? Okay. All right. So it is selectively permeable, allows passage of some molecules and not others. There are enzymes, at least in prokaryotic cells and in the mitochondria of eukaryotic cells, for ATP production. Photosynthetic pigments on infoldings called chromatophores or thylakoids can be found also on the plasma membrane. So here is a photosynthetic bacterium, right? Do photosynthetic bacteria have chloroplasts? What do you think? No, they don't. Chloroplasts are organelles within, and they're strict. And chloroplasts, as we'll learn, used to be bacteria. Yeah, they used to be cyanobacteria, according to the endosymbiotic theory. We'll talk about that in a bit. All right. So these guys are chromatophores. These are basically little folds inside. They're not quite membrane bound. They're infoldings like this, and they're full of photosynthetic pigments, which I'll draw in green. Why would I draw photosynthetic pigments in green? Because a lot of them are green, yeah? And so they're sitting in our, bound in the phospholipid bilayer so that when light hits, they can absorb it and photosynthesis can happen, yeah? Good? Okay. Damage to the membrane by alcohols, quaternary amine, polymyxins cause leakage of cell contents. So if we can target the phospholipid bilayer for certain kinds of proteins, polymyxins are antibiotics. I think my son is on polymyxins right now, and these are things that disrupt the plasma membrane of bacterial cells. Don't we have plasma membranes? Our cells definitely do have plasma membranes. So taking polymyxins orally, we would expect significant side effects. He's taking them as eye drops, and our eyes have layers of protective proteins on the surface before we ever get to a phospholipid bilayer. Okay? All right. So, inclusions, there's a lot of different things besides photosynthetic pigments that we can put in an inclusion in a membrane. We can put carboxysomes, we can put blobs of sulfur, we can put blobs of lipids, we can even put iron in there, and when we do, we have what are called magnetosomes. These are kind of interesting. If you get some rotting sea material and put it in a big jar, does that gross you out? So you can take a jar of seawater with some rotting detritus, throw that in there, and then tape a magnet to the side of the jar. You'll have magnetotactic bacteria that will swim up and land on the surface of the jar next to that magnet. You can get a swab and wipe them off the inside of the jar, and then you can look at these little bacteria that have little tiny magnets inside them. Isn't that neat? So these magnets, the reason they have them is because that helps them detect Earth's magnetic field, right? The Earth is a very gigantic magnet. That's why a compass works, yeah? Anyway, so these guys can use that to navigate to find the environments they like. Okay. Nucleoid. So, nucleoid, what do you think a nucleoid is? So, do prokaryotic cells have a nucleus? They don't have a nucleus per se, but they do have DNA, right? So the nucleoid is where prokaryotic DNA is. Prokaryotic DNA can often has a particular region inside that cell. That region is called the nucleoid, but it's not a membrane-bound structure that we call a nucleus. So we give it a sort of a different name. Does that make sense? Okay. So living things are complex. This is a picture of a eukaryotic cell, and it's got a nucleus, and it's really complex. It's got all kinds of organelles inside. But prokaryotic cells are still very complex. And in order to make a eukaryote or a prokaryotic cell, you need DNA, right? The DNA is the blueprint to make all of that stuff. And so all of your DNA is your genome. Yeah? Have you guys heard that term? A genome is 3 million base pairs for a typical E. coli genome. The human genome is 2,000 times bigger, 6 billion base pairs. A gene is one particular sequence. Bacteria have about 5,000 genes. We have about 17 to 25,000 genes. And again, when we want to make one cell into two, we have to copy that DNA. Yeah? Guys with me here? We'll talk more about all these processes later. But these are terms I want you to be familiar with. Gene and genome. And then a chromosome is a single molecule of DNA. Yeah? One piece of DNA. And in bacteria, the place where that piece of DNA sits, they generally have one piece of DNA, and that's the nucleoid region. Bacteria also occasionally have extrachromosomal DNA. You understand extrachromosomal? That's extra DNA that isn't part of that one big chromosome. And those little extra bits of DNA we call plasmids. You might say bonus DNA, right? They carry genes for antibiotic resistant or a pilus, important non-essential genes. Do you guys remember what a pilus is? It's a tube through which DNA can be exchanged from one bacterium to another. Yeah? You guys with me? Okay. So, look here. We've got a bacterium. He's got a plasmid. He makes a pilus. Again, they fall in love and get married, and then we have all of this shenanigans. And then, what kind of can we think about? What kind of DNA is on this plasmid? What kind of DNA is on that? The plasmid, what color is the plasmid? Blue. What's the red part? That's the chromosome. That's the in the nucleoid region of that bacteria. Yeah? Are these two bacteria the same species? Probably, but they don't actually necessarily have to be, right? But this process is more logistically difficult the more different they are, but it can still happen. All right. So the question is, what kind of DNA is on that plasmid? Is the information on that plasmid essential for the life of the bacterium? Yes or no? No. How do we know that it isn't essential, Michael? The other one doesn't have it. And if he didn't have it and he was still alive, then it would not be necessary for life. You agree? Everyone get that? Okay, so it's not, whatever it is, the bacterium's not going to die without it. Or at least not going to die straight away. Yeah? Okay. So, what kind of information is on there? Whatever it is, it's not essential for life. What do you think? Is it DNA? So the plasmid is DNA, right? Right? But it's not the code to make DNA. It is DNA, yeah? What kind of information? DNA is the code that helps any living thing make proteins, yeah? What kind of proteins are being made by a cell now that it's got the plasmid? I think this is kind of an interesting little quirk here. He's making a pilus, yeah? Was he making a pilus before? No. What kind of information do you think is on that plasmid, Michael? How to make the pilus, yeah? Does that make sense? This guy has the plasmid and he can make the pilus. He gives the plasmid to this guy, suddenly this guy can make the pilus. What kind of information was on that piece of DNA? Looks like it was the information for how to make that pilus. Does that strike you as kind of weird? I think this should be strange to you in a way.
Like how would you strange that it just seems like, okay, well, cool, now the other one has it.
How'd the first one get it? Well, it must have done the horizontal prokaryotic hokey pokey with another bacterium, yeah? Is this recording? Prokaryotic horizontal hokey pokey. I'm going to get fired. Anyway, sorry. So, anyway, so the thing I find unusual about this, what's good about having the plasmid? You can replicate yourself, can you? What do you mean by yourself, Michael? You can transfer your information to somebody else. What do you think? Is it your information getting transferred? It's coming from you, I guess. Insofar, as long as you consider that blue plasmid you, then yes.
It started with another bacteria.
So, who benefits from this transaction? You think benefits is the plasmid. So there's a guy, a pretty famous guy. Most of what he does now is argue with people. There's a guy named Richard Dawkins. He wrote a book called The Selfish Gene. Where's the information for the pilus? It's on the plasmid, yeah? And how does the plasmid get spread? Through the pilus, yeah? Right? So this is to some degree, this is a self-interested little gizmo here, isn't it? The only thing, right? Is this bacterium on the left superior to the bacterium on the right? Is he better adapted for survival? That's about that. I mean, the only thing this guy has is the ability to give this piece of DNA to somebody else. This is kind of like the bacterial equivalent of a chain letter. You ever get those emails that say, happen, and if you don't deliver it to 20 other people, bad things are going to happen. Don't you love getting those? Don't you want to punch the person who gave it to you, yeah? You know what I do is I send the same email back to the person who sent it to me, and if it tells me to send it 10 times, I sell it. And now that person has to send it to somebody else, 100 different people, I guess, because they got it 10 times. Actually, I did that. I had a great amount of fun doing that one time, and then it was such a hassle explaining why they had to send it to 100 people and so on. But anyway, that I just ignore them now. But anyway, so what makes the chain letter spread, right? Well, the chain letter spreads because the chain letter tries to make you spread it, yeah? What about this? Looks like this thing is Are robots going to take over the world? In order for robots to take over the world, what will robots need to do? Think for themselves, and so I had two different answers. Think for themselves and reproduce. What do you think? Which of those? So self-replication. Who can do this? All living things can do self-replication, yeah? Right? What about what about robots? Not yet, right? And if they can't reproduce themselves, well, then we probably shouldn't worry as much about them. They're taking over the world. But once they learn how to make themselves, make more copies of themselves, conceivably, and if they don't make themselves the same way every time, then they're evolving, aren't they? If they make slight differences every time they replicate themselves. Anyway, I don't know if that's useful, but I think it's a pretty. Does anyone find what I'm discussing interesting? Do you see a connection between robots taking over the world and this plasmid making this pilus? Okay, so there's one other thing before I leave this slide that is important about plasmids, and that is that sometimes they carry the gene for a pilus, as I say here, but sometimes they carry other genes that are also not essential for life, but they're pretty helpful in a pinch. One of them is genes for antibiotic resistance, yeah? Does this matter? Antibiotic resistance is a problem. Can you see how having antibiotic resistance on a plasmid is dangerous for people, yeah? Why? Because plasmids can be spread more easily, right? This is a little chain letter. If your little chain letter says, share this with 10 friends, if that gave you the blueprints for how to make a weapon, right, then everybody who got the chain letter could make the weapon. You agree? Well, if all those people who got the chain letter were your enemies, right, then you, as the enemy of those people, would be in bigger trouble when they all had weapons. Would you agree? All right. Well, is antibiotic resistance a weapon? Is it a weapon for your enemies? Insofar as pathogens are your enemies, and if you're a nurse, then pathogens are the enemies of your patients, yeah, aren't they? So this is how antibiotic resistance can spread through little molecular chain letters sent from the inbox of one bacterium to the inbox of another bacterium through the pilus internet, so to speak. Okay? Is that clear? we'll see that often as we go, or you'll see it again as we go through the class. Questions? Okay, so I hope that was interesting. I think it's interesting, and that's why I talk about it so long. And talking takes time, and I'm looking at faces that look more tired than they were last time I checked in with them. Sorry about that. All right, I'll try to wrap up this, and then if you guys need a break, we can take a break, okay? Okay, so structure that regulates passage of substances into and out of a cell is plasma membrane. Great, okay. Nicely done. So, next topic, ribosomes. They are the site of protein manufacture. Proteins. The blueprint for making proteins, where is that? DNA. The proteins themselves get built at the ribosome, okay? So here we can see a piece of messenger RNA, and we can see the individual protein coming out of this blue and beige thing that is a ribosome, right? Ribosomes have two parts, and again, we can see the same thing here. This green thing is running along a tightrope of messenger RNA, and coming out of its head is this yellow glob of protein. Okay. Ribosomes are slightly different in eukaryotes, but the function is the same. Because bacterial ribosomes are different, they can be a target for antibiotics. And we'll talk about that later on. There's ribosomes. Do you see where they are in this picture? They're the little red dots, yeah? Okay. They are submicroscopic. You can't see ribosomes when you look in the light microscope. However, we can see them when we look in something called an electron microscope. Okay, so here, transcription of DNA, translation of DNA. Translation happens at the ribosomes. So here is the protein. It says polypeptide here. If you want to write protein in parentheses next to that, I think that would be instructional for you, okay? A polypeptide is one subunit in a protein that has quaternary structure, yeah? Why do we have the word polypeptide here? Well, what are these little purple things? If this is a protein, what are these little purple things? They're amino acids. Good, right? So a string of amino acids, isn't that a protein? Remember, proteins that have quaternary structure are groups of chains of amino acids stuck together, right? So this is maybe one of those groups, yeah? Guys got it? Or one of those chains, rather. All right. So here is your first introduction to ribosomal math. There isn't a whole lot to it, but I think you'll find it entertaining or frustrating or ridiculous or whatever, but here it is. This ribosome is called a 70S ribosome. It's made out of a 30S subunit and a 50S large subunit. Those two together come together to form the 70S ribosome. Yay, ribosomal math. Don't you love it? Okay. Do you need to have an explanation of this, or are you good with ribosomal math as it is? Explanation, please? Okay, so the S stands for Svedberg units. In other words, sedimentation rate. And basically, Svedberg units are a measure of how quickly something settles, yeah? And so, so basically, I'm trying to think of a good analogy. So this is how quick. Do you guys know what a centrifuge is? You cut yourself. What comes out? Blood. What's blood made out of? Plasma, red blood cells, white blood cells, platelets, yeah? They're all mixed together, yeah? Is there any way you can fish out the red blood cells and separate them from all the other things? spin it around. How many people are familiar with centrifugation as a way to separate out the constituents of blood? Yeah? All right. Well, how long do you have to spin it for? If you spin it for 5 seconds, will you have red blood cells at the bottom? Depends on how fast you spin it, I guess, huh? For those 5 seconds, right? So, the time it takes at the speed you spin it is called its sedimentation rate, right? Do you guys remember why the red blood cells sink to the bottom? They're heavier. They're denser, and that's because they have iron in them from hemoglobin, and you get the high five because you remember that from my class. So thank you. When people demonstrate that they've learned, I appreciate that. So thank you. Right? So because they have a natural tendency to sink, but it's only the spinning that brings that natural tendency out. Does that make sense? Right? All right. Well, ribosomes can be separate, or they can stick together as a functional unit. Right? When they stick together as a functional unit, that unit sinks to the bottom when you spin them around in a centrifuge. If you take cells, right, and instead of centrifuging out cells, you're now centrifuging out organelles, structures inside them, yeah? So you crack open the cells, and now you have a goop filled with lysed cells, yeah? Broken up cells. If you spin those cells around, big ribosomes will sink faster than the pieces, right? This piece together sinks at 70 miles an hour, so to speak. This one sinks at 50 miles an hour. This one sinks at 30. Does that make sense? Are you satisfied with ribosomal math now? Does that make sense? Sort of? If you can articulate a question, I'll try to answer it. Do you have to do the math in the test? You should. That's a good question. You definitely don't need to know the math in the test. You should know that prokaryotic ribosomes are 70S ribosomes. As we'll learn in Chapter 4, Part 2, eukaryotic ribosomes are 80S ribosomes, okay? Both kinds of ribosomes are made of 2 pieces, a big piece and a small piece. Both of them show the same ridiculous math, right? But the only thing that you should know is that the ribosomes of eukaryotes are slightly different. And one way we characterize the difference between prokaryotic and eukaryotic is by saying prokaryotic are 70S and eukaryotic are 80S. Okay? Guys good? And hopefully that's a satisfactory explanation of what you guys have to know. Yeah? Okay. So much for ribosomes. Now, cytoplasm. You guys know what cytoplasm is? It's the fluid inside the cell, yeah? Inside that fluid, there's proteins floating around in there. There's water that makes up the most of it. There's salts dissolved in there. It is the aqueous medium for the chemical reactions that happen inside the cell, yeah? Guys, understand what I'm saying there? Okay, good. All right. In other words, the stuff inside the plasma membrane where the organelles float and where the chemical reactions happen. Okay. Lastly, endospores. All right. So endospores are unique to bacteria, but they're not just unique to bacteria. They're unique only to a very specific group of bacteria. They're only found in certain kinds of gram-positives. Not all gram-positives, just a few genera. Genera is the plural of genus. You guys remember genus and species? Right? Bacillus is a genus along with a shape, right? Bacillus and Clostridia are the only two that, I'm pretty sure they're the only ones that make spores. And endospores are spores that are made inside a cell. We can see here that they're stained a little differently. They're stained with something called a Schaefer-Fulton stain, and that requires malachite green. And malachite green has this sort of green color. And you can see that these spores are occasionally floating out by themselves, but a lot of them are sitting like little bubbles inside the little bacteria. Can you guys see that in the picture? guys see that way the heck in the back? See that okay? Right? So these things sit inside the bacteria. The spores, do you understand what they are? They are basically little dormant, tough, protected, sleeping cells. Yeah? Dormant, dormir, to sleep. Yeah? In Portuguese or Spanish or French, Latin languages. It's a Latin-based word. Yeah. So these little guys can't eat. They can't grow, but they can wake up. And while they're dormant, they are tough. They're hard to kill, and they can last a long time. Right? So, do you guys ever see the movie Austin Powers? Right? Remember Dr. Evil? How did he manage to show up? Remember he's floating around in space in that little egg going, it's freezing in here. Do you guys remember that? At the end, and he's sitting there chattering and so on and so on. It's just cold all the time, right? That's kind of analogous, right? He's still Dr. Evil. Once he gets back down to Earth, he can still do all the nasty stuff, right? But while he's up there, he's protected because he's out in space. He's not harming anybody because he's in this little egg shivering. Yeah? Is that clear? How many people have not seen Austin Powers? Okay, so I feel like I've wasted your time, and I'm sorry about that. Should they see Austin Powers just in order to understand this concept?
Yes.
Well, yeah, so you could probably learn about endospores more efficiently another way. Anyway, so anthrax endospores, this is Bacillus anthracis. We are more interested in these because they're a potential bioweapon. Are there other spores that we care about? Well, Clostridium difficile. Have you guys heard of C. diff? Right? Clostridia form spores, and those spores are hard to kill. Clostridium tetani, it's the spores that sit on the tip of the nail when you step on the nail and you can get tetanus. It's because the spores get implanted deep inside your body, and when they're deep inside your body, they wake up and start wrecking up the place, yeah? And causing tetanus. Yeah? Yeah? Palms. Oh, yeah. So that's a great and important distinction. Endospores are unique to bacteria. Only bacteria have endospores.
No, I know, but it will be kind of like the same. There are like little things sticking in the bottom of the leaf, and then the things fall off the leaves.
Yeah. So a seed is analogous. It's not the same, and there are other kinds of spores that exist in nature. Mold makes spores, right? Mold spores are different though. They are reproductive. I'll go back here. How many bacteria here? One bacterium. What does he do? He copies his DNA, he pinches himself off, and then he bursts and his spore is released. Yeah? How many bacteria to start with? One. How many bacteria to end with? One. What happened to the bacteria that this thing hatched out of? It's dead. Yeah? So, the numbers of bacteria do not increase when a spore is formed. Right? So this is not a this is not a mechanism of reproduction. What about the pollen of a tree? Pollen of a tree is reproductive, right? And it floats around, and it has to. Pollen is different from spores because pollen needs to fertilize an egg, right? Pollen is much more like a little sperm than it is like a spore, right? Whereas mold spores, those can float around from, you open your bread, right? You take out a piece of bread and you see there's a bunch of mold on it, right? And the mold is all powdery and dusty. And you drop the bread and a little cloud of spores goes out, and you know that the rest of your bread in the house is all going to get ruined because you just released a bunch of spores, right? Each of those little guys is ready to start growing on his own. He doesn't need to find a mold egg in order to start growing, right? A plant that has pollen has to have that pollen find a seed in order to fertilize that seed to make a new plant. Does that make sense? So it's not a gamete like, in other words, a sex cell like an egg or a sperm in an animal, or a pollen grain and a seed in a plant. It's not reproductive like a spore of a mold where each little guy can make a new critter, and zillions of little guys can come from one mold creature. Yeah? Does that make sense? So that mold, when you drop that bread, you're going to pick up that bread, you're going to throw it in the trash, and that piece of bread is going to go out to the landfill, and it's going to be able to spread to all the other stuff in your trash, and it's going to be able, once that bag of trash gets out to the landfill, there's, the bag of trash is going to be broken, and those spores are going to float out, and they're going to go and spread far and wide across that landfill and reproduce on all the other goodies that those spores happen to land on. Does that make sense? So that's reproductive, where the individual that made the reproductive spores stays alive. Yeah? So reproductive spores can radically increase the numbers of a particular creature, like a mold, right? These guys, they don't make the bacterium reproduce, but they make the bacterium very, very hard to kill, right? Does anyone work with patients right now and have the problem of C. diff come up? Clostridium difficile, right? And what a huge hassle. If somebody mentions the word C. diff to you, you probably get a headache and groan, and you're like, oh my God, we got to clean all this. We got to move all these patients from this place to the next place. We got to do this, that, and the other thing. And why is that? Well, because getting rid of C. diff spores, once they get out into the environment, is a huge, huge headache, because they are tough and they survive. You can carpet bomb with bleach and other chemicals, and that'll usually kill them. But if you get one little guy, then you didn't kill them all. Yeah? Does that make? Am I making sense here? Right? All right. So hopefully this next little tidbit will provide an illustration of what makes endospores so tenacious. So how long can endospores remain dormant? Microbe world scientists, profiles, yada, yada, yada. Hopefully this comes up. Boom, here we go. Don't use recommended settings. Okay, there is Internet Explorer. No, that's not what I, there we go. That's what I want. This guy, he looks kind of nerdy. I mean, if someone was taking my picture, I'd send him a better picture than that. Anyway, so here he is, Raul Cano. He's a professor of microbiology at Cal Poly San Luis Obispo. You guys know where that is? How many people know where San Luis Obispo is? I've been there a couple times. It's nice. It's right on the coast. You get sea breezes. It's north of Los Angeles. You don't have to deal with all the pollution. Nice place to be. I haven't spent too much time there, but I envy those who live there. Anyway, so he gets to live there, and he and his colleagues in 1995 stunned the world when they announced they'd revived 30-million-year-old bacteria from spores taken from the gut of an ancient bee entombed in amber. Did you guys ever see, what's that? Yeah, Jurassic, whatever you call it, Jurassic Park. where they get a mosquito and they take cells from a Tyrannosaurus rex out of the gut of a mosquito and reconstitute that Tyrannosaurus rex, right? Maybe not so realistic because the red blood cells and white blood cells, rather, of the Tyrannosaurus rex do not have an endospore coating, right? They are vulnerable to the digestive juices of the stomach of the mosquito, right? We can expect that DNA will have been chopped into tiny little itty-bitty pieces, right? And will be so much the more difficult to reassemble. But if we have a little endospore, that can stay around a little longer, yeah? And how much longer? 30 million years. 30 million years old. Isn't that crazy? Whoa. Anyway. All right. So that's all I got, I guess, huh? Some bacteria are gram-positive, some are gram-negative because of differences in their cell wall. Correct. All right. What do we say about endospores? Two is the only correct answer, right? Makes them tough, makes them survive environmental changes. All right. So that's all I got. Do you guys want to take a break?